Thank you for the introduction. Humans are very good at certain types of pattern recognition tasks, particularly vision-based tasks. But you get increasingly unreliable when you ask them to do tasks repetitively. Computers, by contrast, are very good at repetitive tasks, but require precise training to be any good at them. So the question that we were asking is whether we can find algorithmic measures for evaluating image quality that are equal to the human ability to evaluate these imaging measures. And so image quality is obviously paramount importance for any analytical data. You face the garbage in, garbage out problem. And this is just as true with image analysis-based analytics. And so just to illustrate how good humans are at pattern recognition, if I show you these two images, I'm sure you'll agree that the image on the left shows some structured patterns, whereas the image on the right does not. And despite none of you being mass spectrometry experts, you've correctly picked out a distribution that corresponds to a peptide that's localized with the neural bodies in the rodent brain. However, when you look at more challenging data, and this particularly occurs when you're looking at low abundance molecules and you're pushing against the signal-to-noise ratio, judging whether images are of useful analytical quality becomes a little more challenging. And at this point, we'd sort of turn to domain experts with a lot of experience in analyzing these, interpreting these images to judge whether these images are of sufficient analytical quality. And these both show distributions of low abundant peptides. So to back up slightly, because I suspect not everyone here is very familiar with imaging mass spectrometry, I'd just like to give a quick introduction to the technique and to the image formation process, just to explain why we need to reproduce these quality assessments algorithmically and the scale of the problem that's faced. So the measurement process starts with taking a sample, and you collect a thin section from the sample. We coat it with a matrix that's an energy coupling factor. So a little bit of sample prep, and we then interrogate a certain point with a laser. This laser causes a small explosion at some point on the sample, and the material from this explosion is then transported to a mass spectrometer where it's analyzed. And essentially the mass spectrometry separates out the molecules that are from that point in the sample by separating them according to their mass. And so, I don't know if we have a laser pointer. Every peak within the spectrum corresponds to the abundance of a molecule of a particular mass to charge value. And so the different peaks correspond to different chemical species, and the intensity of the peak corresponds to the amount of that species that was detected with the mass spectrometry. And you can see here, if you collect spectra from different points on a sample, which is got color-coded here, you get very different spectra according to the composition of the sample at that location. And so we build this up into an imaging modality by collecting spectra at every point across this grid. These get then combined together into a hyperspectral data cube. And so every pixel here contains a complete mass spectrum in this X and Y dimension. And then along this chemical axis here, you have the intensity of all of these points. And so if you slice the cube here and plot the intensity of a particular peak over the image, you end up with the chemical distribution of that molecule within the sample. And within these data sets, most of the channels correspond to some kind of noise. So very few actually show typically very structured distributions. So even though you have somewhere between 50,000 and 1 million individual mass measurements, there's typically more like a few thousand sort of useful analytical images in there. And so the typical way of finding these is you get a mass spectrometrist and they click through every single image in the data set, determining whether or not there's a useful distribution there. And obviously this isn't a particularly pleasant process for the mass spectrometrist. It's time consuming and it delays the speed to any sort of analysis of the data. So the challenge is really finding the high quality images within these data sets, and particularly the low abundant images that are still of useful analytical quality, but aren't necessarily of very high sort of absolute quality. The question is really where this threshold for quality occurs, whether people agree with each other on what this threshold for quality is, and then whether we can find this algorithmically. So the first thing we need to do is to build a training set so that we have something to compare algorithms against. And so for this, we had some ideas personally of what we looked for in our images, but we were never really sure if it was the same thing everyone else was looking for. So we wanted to get some kind of community consensus on the quality of images as the precursor to anything algorithmic. So we turned to the imaging mass spectrometry community. We devised an online survey for getting their input on image quality. And we recruited as many experts as we could to help us to annotate these data sets to create some kind of gold standard for use in the algorithm training. So after some preliminary work, we came up with a pairwise comparison as being the most approachable method for getting this data. So we showed experts a series of annotated pairs of images. We showed experts a series of pairs of images. They then annotated the relative quality by moving this slider bar towards the image that they thought was of the highest analytical quality. And this let us really show these images to experts without having to train them on an absolute scale, without getting them involved in a large, time-consuming ranking exercise. And it gave us a lot of high-throughput judgments on these images. So we took a set of images that were sort of semi-randomly selected from several of these hyperspectral data sets. These data sets were generated with some variation in instrument parameter, so they gave us a bit of a spread of image quality. And then to avoid just having every image being complete noise, we had put some bias towards the intensity. But in general, we collected a set of generally pretty low quality looking images and then presented these to the experts to get their feedback on the difficult cases. And so we took 52 images. Of these, we took 50 and generated every pairwise combination of them. We then reserved one pair of images to use as a consistency check, which I'll describe in a little more detail later. These images were then, experts were then recruited to the online survey and shown a series of these images. So every expert ranked 49 at a time. It took us about 4 months to get enough people to take the survey, which we emailed basically everyone we knew, recruiting them to this, asked on LinkedIn, tried to put the message out there as far as possible. But overall, we got generally people that had more than 3 years experience on average, and a total of 260 years of imaging mass spec experience. So we felt we really covered the whole field with who we recruited here. So even just to annotate these 1,200 odd pairs, it took over 12.5 hours of human time. And we were looking to get 3 ratings per pair, so we'd have some stats to work with. So we needed to triple the number of ratings that we had image pairs. It took an expert about 10 seconds to make a decision. So these were kind of snap, intuitive decisions, which is what we were looking for. So I mentioned we had this pair, we kept one pair as a consistency check. This pair was shown to every expert 3 times over the course of the survey. And if they weren't able to be self-consistent with their response to this pair, they were then excluded from further analysis. And this cut about a quarter of the people that took the survey out. So we were left with 60 raters that successfully completed the survey. And so this left us with about 600 pairs that had the 3 ratings that we needed. So the first question is, did the experts even agree with each other on the quality of these images? We know we'd chosen some fairly challenging pairs, and particularly making this whole combinatorial, every possible pairwise combination really generated some challenging pairs. So there's a measurement called Krippendorff's alpha, which measures the disagreement in the ratings that experts give, parameterized by just the expected disagreement by random chance. And something above 0.6, 0.66 is considered sort of acceptable for this sort of human study. Of those 600 pairs, we had an agreement of about 0.5, which was a little low. But we hypothesized that probably some of the image pairs we were giving were just kind of tough. And it was just not possible for the experts to really give a consistent opinion on them. So this takes a little bit of explaining, we took each pair out one at a time and recalculated the overall agreement. And what we found is there's a small number of outliers down here that when you include them in a data set, really negatively impacts the overall agreement. So there are a small number of images that have a disproportionate effect on the cohort agreement. So we systematically removed these one at a time based on how negatively they impacted, and then recalculated the agreement for the remaining set of image pairs. So the guys down here disproportionately negatively impact the agreement. So we take them out one at a time. And what we see is that the overall agreement steadily increases to some plateau, and then the measure starts to break down when there's a small number of pairs left. And so only taking out about 20 images, we're up to this sort of 0.6 threshold that we need for sort of to say we've got decent agreement between our raters. And then taking out 100 or so, and we're up above 0.7, and then we max out at 0.85, which is a very high agreement for these sort of human type studies. So based on this, we defined 3 sort of levels of gold standard. So we had sort of the most challenging set, which is where we had sort of the minimum acceptable level of human agreement, and then a further 2 where we had increasing levels of agreement, and these should be easier and easier to match against the experts for. So we took these gold standard pairs and we evaluated a whole bunch of image-based measures of potential measures of quality. And so because we didn't know what people were looking for when they evaluated these, we sort of trawled the literature to look at all the image-based stats we could come up with. And so most of these will be reasonably familiar, looking at sort of intensity-based measures for the histograms, and then sort of local statistical properties. And then finally, this measure spatial chaos, which we recently published, which uses structure within the images. For the intensity-based measures, we then calculated a few summary statistics over the whole image. These were calculated in a patch-based fashion, so we took a moving window over the image and then calculated these summary statistics for them. And so between all of these combinations of image descriptors, we have 167 potential measures of quality, 167 image features. And we compared these against the ratings that the experts provided, and we calculated the linear correlation, and then also whether or not they correctly classified that left image was better than right image as sort of a binary classifier. So the measure that might be unfamiliar is this measure of spatial chaos, which came out of our lab a couple of years ago, where we look at structure in the image using sort of a 3-step edge detection and then nearest neighbor graph calculation. And this says how closely connected all of the structured pieces of the image are, and this gives us a single number that describes the overall structuredness of the image. And this is the motivation behind doing this. And there's more details in the paper there. So we took these 3 levels of gold standard, we calculated the correlation against the expert ratings, and also this sign match sort of type classification. And we found that actually even on the worst performing data set, we were able to get an accuracy in terms of whether left was better than right of greater than sort of 0.75, reasonably strong correlations. And when we went to the very, very high agreement gold standard set, we got almost very high accuracy and a correlation of greater than 0.9. So these measures individually, some of them were able to very precisely reproduce the opinion of the experts on these image pairs. Unfortunately, our measure wasn't the number 1 like we were hoping, but it was in the top 3 out of this 167, so we were still reasonably pleased with that. And the top performing ones were standard deviation based on a sort of medium scale patch as a proportion of these images, and it's the maximum absolute deviation of those measures. So it seems like people are looking for some kind of measure of the local variance within the images as the primary thing they're using to assess these. We also tried some machine learning on combinations of these, but we didn't really see much improvement over the individual measures. So we now had some measures that we felt stacked up pretty closely to what the experts were thinking. And so we want to use these to replace this sort of manual data trawling part of the analytical process. So to determine which images within a data set are of sufficient analytical quality. So to do this, we took the top performing algorithm, this MAD STD, and then applied it to every possible image within a data set, We then ranked it according to this quality score, and you end up with this fairly characteristic curve where you have high-scoring images on the left, and then there's not that many of them as a function of the data set. And in total, there were about 6,500 imaging channels in this particular data set. And so what you can do is get an expert to then very quickly slide down this curve, look for the point at which the image is stop being analytically useful, and you know that everything else to the right of that image is just going to get worse in terms of quality. So this takes a couple of minutes to calculate on a data set compared to the maybe 20 hours that it would take for an expert to go through 6,500 images. And then it's very simple to determine the analytical cutoff, and then only those images that are analytically useful are then go on to be used for the biological interpretation. So to just quickly summarize, we did successfully reproduce the human judgments. Humans were able to agree with themselves, which was a pleasing finding in itself. And all of this code, all of the images, all of the survey results are available on the GitHub. So if you want to reproduce this analysis, if you think you have an algorithm that might perform better, you're welcome to go and download those and have a go. And then I'd just like to finish by thanking everyone that was involved with this project, the laundry list of institutions, but particularly Katya, who helped us so much with putting this survey together, and the data collection that was done at the University of Benz, and also to the ISMB for the Travel Award that allowed me to be here to talk to you today, and to you for your time. So thank you.